Okay, so welcome back everyone. We should get started. So this week we're talking about the anatomy of the brain and the research methods that are used in biological psychology to understand the function of the brain. To introduce the topic, I thought it would be interesting to show you this video that was posted on YouTube by a man named Andrew Johnson. And you'll see in the video that he has a kind of early onset form of Parkinson's disease. And you may know that one of the main symptoms of Parkinson's is this loss of motor control. So that can take the form of tremors of the hands and the body, for example. And this occurs following the loss of neurons in a part of the brain called the basal ganglia. And so these are neurons that produce dopamine and seem to have a role in controlling movement of the body. What you'll see in the video is Andrew demonstrating this kind of extreme treatment for Parkinson's disease called deep brain stimulation, which is where electrodes are inserted permanently into the patient's brain and apply electrical stimulation to the basal ganglia region. And you'll see that this can have quite a profound effect on a person's symptoms.
Hi, I'm AJ. I'm 39 years old and I suffer from Parkinson's disease. I was diagnosed 4 years ago when I was 35 and I've since undergone deep brain stimulation surgery. This is a procedure whereby two probes are dropped deep into my brain and hooked up via a wire down my neck to a pacemaker in my chest. This provides a steady stream of electricity to my brain per second. So it has had a major effect on my life and a major effect on my family's life, which is the main thing. And today I'd like to show you what happens when I turn my neurostimulator off. Now, it's just after 11 AM, so it's time that I had my second dose of medication for the morning. So my first dose is pretty much all but worn off. So this is a good example of what I'd be like when I'd be sort of off, with no medication in my system and no power. To show you the dramatic effect, I will demonstrate. This is me with the power on. I'm not making happy little faces for the video. These are tests that the neurologists make us do to test our reflexes. As you can see, they're wiggling, moving fine, and I've got very good movement. This is the remote control, and this checks that I'm on. On and okay. And this is me when I turn it off. You won't be able to read that, it's shaking too much. So it's almost an instantaneous reaction. I can't control this. It shakes some quality cocktails with the right. The left just looks like the royal wave. My voice is gone. It's harder for me to get words out and speak. I'm completely rigid, and if I tried to stand up I'd fall. If anyone thinks I'm faking this, I'm not. I can't control the tremors and dyskinesia. The twisting in my neck is called dystonia and is really quite uncomfortable. So as you can see, the tremors are getting much worse and I'm in quite a bit of discomfort. And I'm about to drop the remote control, which will be fun because I won't be able to turn stuff back on. So I think party time is over. Almost automatically. The power comes back on. I'm steady. So if anyone's considering DBS, I thoroughly recommend it. It is life-changing. Thanks.
Okay, so a pretty amazing video, I think. And to say a bit more about the technique, what it involves brain surgery, where the patient has their skull opened up and these electrodes inserted deep into the brain to come in contact with the basal ganglia area. And in particular here, these electrodes are targeting a region called the subthalamic nucleus, or STN, which is here. And the idea is that the patient just goes about their life every day with these electrodes that are connected to a kind of pacemaker of sorts, applying a weak electrical stimulation to the neurons that are found around this region of the brain. And that electrical stimulation of neurons seems to go some way in many patients towards counteracting the loss of function caused by neurons dying in this region. So the reason that I mentioned this at the start of today's lecture is that it's an example of how we can link the function of a particular region of our brain to a particular kind of behavioral or cognitive function. And so the fact that stimulating this STN region has such a profound effect on Andrew's symptoms is quite vivid evidence that this region of the brain has some role to play in how we regulate the movement of our body. And so in this lecture, we'll talk about how we can divide the brain into different regions and then, to some extent at least, link those different parts of the brain to different functions of our minds and our behaviour. So we'll also talk about research methods that are used in this area to uncover the function of different regions of the brain, and that includes methods like deep brain stimulation, as well as methods used to visualize the brain and create images like what you see on the right here. So for the first part of the lecture, I'm going to talk about research methods, and then in the second part, the anatomy of the brain and the nervous system. By the end of the lecture, you should be able to describe the main methods for studying the relationship between brain activity and behavior. And I think the most important part here is being able to contrast the strengths and limitations of those methods, which affects the kind of research questions that we can use these tools to address. And then the point of this lecture is also to familiarize you with the terms that are used in this field to describe the anatomy of the brain, and to give you the ability to give an overall picture of or description of the gross anatomy of the brain. And so by gross, I mean macroscopic, as opposed to the microscopic features of cells that we've been talking about in the previous lectures. So starting with how we can study the anatomy of the brain, there's actually quite a lot of different techniques, and I won't go through them all, but instead I'll focus on a few key techniques that are used very commonly in the field. And you'll see that these different techniques can get at different types of anatomy, so the anatomy of individual cells versus the anatomy of the brain as a whole. In the first lecture, we talked about how the basic structure of a neuron was first described by Spanish researcher Cajal, who was the first to look at neurons in detail through a microscope. And this discovery, his work, relied on the development of what's called a staining technique, in particular something called the Golgi stain, which is still in use today. And so what this involves is taking a piece of brain tissue, and so in the photos here is the brain of a mouse, and then soaking that brain tissue in a chemical that is able to stain and change the colour of some of the neurons that are present in that brain tissue. And then the tissue can be cut into thin slices, mounted on a slide, and then looked at through a microscope to get images of the kind of fine scale structure of the neurons, like this. A feature of the technique is that not all of the neurons that are present in the tissue are stained. And so this tissue actually is much more dense with neurons than what you see here. But the staining technique only affects the coloration of a certain subset of those neurons. And that's what really makes it useful, because if all the neurons were stained black in these images, you wouldn't really be able to see anything. It would just be a tangled mess. But because it's only affecting a subset of the cells, that allows us to see the fine-scale anatomy of each cell more clearly. A more recent technological development is electron microscopy. So on the left here is an electron microscope, and this can produce even higher resolution images of individual cells, so much so that we can see subcellular structures, so structures that are found inside the cell, like these vesicles, for example. In the first lecture, I mentioned this recent innovation in how electron microscopy is used, that allows researchers to build up a detailed model of all of the neurons that are present in a particular part of brain tissue. And so in this example here, they're taking a very small cube of the cortex from a mouse. They've created these 2D cross-sectional images of that tissue using an electron microscope, cross-sectional images going all the way through this block of tissue. And then they're able to carefully label where the different structures are in each of those cross-sectional images, and then stitch that information together to recreate a 3D model of all of the neurons that are present. And that provides us with this detailed view of the structure of individual cells, but also how those cells are connecting together, at least within a small part of the brain. Okay, so techniques like microscopes have been around for a long time, but there are still these new developments happening that are allowing us to visualize the brain in new ways. These techniques for visualizing the structure of individual cells rely on samples of brain tissue that have been taken from a deceased person's brain or the brain of an animal. And we can also use these kind of post-mortem samples, so brains donated to science, to study the overall anatomy of the whole brain. So that's what we're looking at here. So on the left here is a view of a post-mortem brain from above, and on the right is a cross-sectional view where the brain is being cut like this, and then again we're sort of viewing it from the top. And you can see straight away that there are a number of different features of the brain's anatomy that we can see just with the naked eye. All these kind of wrinkles and folds in the cortex, and when we're looking at the brain in cross-section, these different sort of lumps of darker matter and kind of tracts of lighter matter. And so we'll talk about what those different anatomical features are a bit later in the lecture. Nowadays, we also have these techniques that can visualize the anatomy of a person's brain while they're still alive. And so the most commonly used of these is MRI, so magnetic resonance imaging. This can create images of the brain like you see on the right here, where we can also see those anatomical structures like the cerebellum here, the brainstem, the cortex at the top. And so it's providing us with a level of detail, a level of resolution within the scale of millimeters, and that gives us a view of the brain that's pretty similar to if we were kind of looking at the brain with our naked eyes. What it doesn't give us is a view of individual cells, and so back here when we were looking at the synapse for an electron microscope, the scale here is in the order of nanometers. The scale in these images of individual neurons is in micrometers. And so the millimeter scale produced by MRI is not allowing us to see individual neurons, but rather just a larger scale anatomy of the brain. This is an example of an MRI image, so there's a link to this on the slide. And MRI creates three-dimensional images of the brain. So here we can see those same structures: the cerebellum, the brainstem, the cortex, and then we can also kind of scroll through the person's brain to look at it from different angles. These are the eyes, and later in the lecture, when we talk about what some of these kind of different areas of the brain are, you might find it useful to come back and see if you can identify those areas in the MRI image. And so how does MRI work? It kind of seems like magic to me that we can peek inside of a person's brain while they're still alive. For those of you who have had an MRI before, you'll know that before they let you anywhere near the machine, they kind of carefully screen you. They ask you lots of questions, like do you have any piercings, do you have any implants, and so on. And the reason for that is that the machine includes a really strong magnet. It's generating a really strong magnetic field that is strong enough that metal that's brought into the room can be pulled through the air to attach to the machine like this. When our body is in that really strong magnetic field, what happens is that atoms, so hydrogen ions or protons in our body, particularly in the water molecules in our body, actually effectively rotate to align themselves with the direction of the magnetic field, which is illustrated here. And you could think of this as being similar to how the needle of a compass rotates to point north, so it's aligning with the magnetic field of the Earth. And then what the MRI machine does is it then shoots a burst of radio waves into the person's brain that knock the atoms out of alignment with the magnetic field. And then when those radio waves are turned off again, the atoms rotate back into alignment with the magnetic field. And when they do that, they're releasing energy themselves in the form of radio waves that can then be detected by the machine. And different tissues in our brain and our body, like skull versus brain tissue, for example, will tend to release that energy in different ways, which then allows us to reconstruct a picture of the different tissues that are inside the body. And so this physics of MRI is really quite complicated and definitely beyond the scope of this course. So you don't need to know these kind of details for the purpose of the assessments, but it's really quite interesting, I think, as well. And so I want to just give you a bit of a taste of it. And I linked a video here that visualizes it as well, if you're interested. Okay, and so we can also use an MRI machine to create a picture of the connections in the brain between different areas. And this is a technique called MRI tractography, or diffusion spectrum imaging. And it uses the same MRI machine, but it measures a different signal which relates to the movement of water molecules within the brain along the axons of neurons. And so what you're looking at here is effectively these different pathways of axons inside of the brain. So the front of the brain is over here, the back of the brain is here. In green, we have these bundles of axons that are running between the front and the back of the brain, connecting different areas of the brain together. And in blue, we have axons sort of running between the brainstem at the base of our brain, near the spinal cord, up to the top of our cortex. So this technique, MRI tractography, can be used to create these really beautiful images of these axon pathways in the brain, and this can be useful for understanding how different areas of the brain connect together and form into networks. So the key learning points here are that there are these neuroanatomical methods that are used to study the structure of the nervous system. Some of these allow us to look at the level of individual cells, while others are useful for looking at the overall anatomy of the brain. And there are these modern technologies like MRI that allow the anatomy of the brain to be visualized in living people. Okay, so once we've looked at the anatomy of the brain and we've maybe defined some different structures that we're interested in, like the cerebellum versus the cortex, for example, we can then ask a question of what is the function of those different areas of the brain. And there's different ways of trying to get at this question. One that's used commonly is to study the effects of damage to the brain. So what happens when somebody suffers damage to their cerebellum? How does it change their cognitive function or their behavior? Another is to look at the effects of brain stimulation. What happens if we apply electricity to the cerebellum and change the activity of neurons in that region? And then another is simply to record brain activity while a person is performing some kind of cognitive task. Like if we're interested in working memory, for example, we can design a task that engages a person's working memory and then record their brain activity while they're doing that task to identify what areas of their brain are becoming active. So to give you some examples, one of the earliest examples of studying the effects of damage to the brain involved studying soldiers who had suffered bullet wounds to their head, but they had survived. And one observation from this work was that soldiers that had been shot in the back of the head, damaging a region called the occipital cortex that we'll talk about a bit later, which is at the back of our head, often became blind. So their eyes appeared to be working perfectly, but they still lost their vision after this part of their brain was damaged. And then a further observation was that the particular location of that damage predicted the kind of blindness that the person experienced. So what's been illustrated here is a soldier who had suffered a gunshot wound to the back of the head, slightly on the right side of his head, and had lost vision specifically in one part of his visual field over to the left. And similarly, there were other cases of people who had suffered damage to the left side at the back of their head who then lost vision on the right side of their visual field. And so this kind of work provides a kind of evidence of that our occipital cortex is necessary for us to be able to see. And then also that there are different parts of the occipital cortex that are responsible for our vision in different parts of our visual field. And we'll talk more about the role of the brain in vision in next week's lecture. Another well-known example of studying the effects of damage to the brain was work by Paul Broca that you might have heard of before. So he was a French surgeon who studied people with impaired speech, including studying the brains of those people after they had died. One example is a patient of his who lost the ability to speak during his life and then couldn't speak for the last 30 years of his life before he died, and then Broca was able to study his brain. And what he found in patients like this often was damage in their brain to a part of the left prefrontal cortex, which is highlighted here in red, and which has now become known as Broca's area. And so at the time, this was quite a profound discovery because it was one of the first instances of being able to link a particular part of our brain to a particular cognitive function, which in this case is language. Nowadays, we have techniques like MRI that allow us to visualize where damage has occurred in a person's brain while they're still alive. And so we don't have the same kind of limitations that Broca had of only being able to study the brain post-mortem. And this, to give you an example of how this kind of research is done nowadays, in this study, they were looking at a group of people who had suffered strokes. And so a stroke is where there is bleeding in the brain that leads to damage of the brain tissue. And they asked these people to perform a kind of emotion recognition task where they were shown images like this of people with different expressions on their face, and they just had to judge the emotional state of the person. Like, is this man terrified? Is he upset? Is he arrogant? Is he annoyed? And so on. And what they found is that people who struggled on this task, who found it more difficult to identify the person's emotion, tended to be people where the stroke had caused damage to the part of the brain, this kind of frontal region of the cortex, which is highlighted here. And so it provides a kind of evidence that this part of the brain is necessary or important for our ability to read the emotional expressions of other people. These kind of studies that look at the effects of brain damage have been really influential in guiding our understanding of the brain, but they're also limited in various ways. So one limitation is that they're limited to the kind of damage that occurs naturally. So if you're interested in studying a particular part of the brain, but there are no known cases of people that have suffered damage to that part of the brain, then this isn't going to kind of get you anywhere. Another limitation is that natural forms of brain damage, like traumatic injury to the brain, stroke, and so on, often cause diffuse damage. The damage likely isn't going to sort of fall neatly in kind of one area of the brain, but will likely affect different areas of the brain. And that means that it's difficult to say then whether a person's symptoms are due to damage to one specific area or to another area, or at least it makes it a bit more harder to make that inference. And so a related approaching research is to study non-human animals and cause damage to the brain, and then study the effects that has on the animal's behaviour. And so that can include removing a part of the brain, like removing the cerebellum, for example, or producing what are called lesions, which are just localized areas of damage in the brain, which might be done by injecting a chemical to a really specific part of the brain to cause damage to the neurons that are present in that area. And so the idea then will be to cause damage to a particular area and then devise a set of behavioral and cognitive tests in order to analyze what aspects of cognition have been affected by that damage and what aspects of cognition have been spared, as a way of trying to understand the function of that brain area. Animal research also has its limitations, of course, and so one limitation is that it is in terms of what we can learn about humans by studying animals. And so, for example, if we're interested in how language development occurs in the brain, then studying animals is probably only going to get us so far. And then another limitation is that it raises serious ethical issues about to what extent it should be permissible to cause harm to animals for the purposes of research. And so if you're interested in the debate about the ethics of animal research in the context of biological psychology, there's a couple of sections in the textbook that give a perspective on that debate. Okay, so another general approach to studying the function of the brain is to stimulate the brain, so manipulate the activity of neurons by electrically stimulating them, for example. We saw this example at the beginning of the lecture of deep brain stimulation, where electrodes are inserted into a particular region of a person's brain and apply electrical current in order to modulate the activity of neurons in that area. This approach to manipulating brain activity is very invasive. It requires opening up a person's skull, which is generally frowned upon, so it's not a common technique in research. It's usually only performed when there's a serious medical reason for this to occur. But this technique is used quite commonly in animal research to study the function of the brain. And so what this often involves is inserting an electrode into the brain that will come in contact with a specific neuron, so one neuron, and then applying electrical current to affect the activity of that neuron. So, for example, induce action potentials in that neuron. And then typically it will involve, for example, getting the animal to perform some kind of cognitive task, manipulating the activity of that neuron and testing whether that is changing the monkey's or the animal's performance on that task in order to understand specifically what the role of that neuron is in some aspect of cognition. And we'll see a couple of examples of research like this as we go through the course. In humans, this approach is rarely used, but instead there are these non-invasive brain stimulation techniques that are quite widely used in healthy human populations. One is called transcranial magnetic stimulation, which is pictured here, where it involves placing this device on the scalp of a person's head, and then when the device is turned on, it generates a brief magnetic field that is able to travel through the person's head effectively and modulate the electrical activity of neurons that are underneath that part of the scalp where the device is placed. And so this is just a short demonstration of this technique.
Transcranial magnetic stimulators, or TMS for short, passes electrical current through the coil to generate a rapidly changing or a constantly changing magnetic field. And when we place that magnetic field on the scalp, we can pass electrical current through the brain. I can stimulate Guido's hand area and basically change the behavior of his hand, so I can make the hands twitch. Okay, and now. Hopefully you'll see his hand goes, the line goes a bit wobbly. That's because this current is going through his brain.
So what's happening there is he's asking the guy to draw a straight line, and then he's zapping his brain with the TMS device, and it's causing this sort of twitch in his hand.
Affecting his hand.
Yeah.
Thanks, Guido.
So to unpack what's happening there, if you imagine this is Ido's brain, the TMS device is placed on the surface of his head, and in this case it's targeting this area called the motor cortex, which is found at the top of our head here. And the motor cortex has motor neurons that have their cell bodies in the cortex and then send their axons down through the brain to the spinal cord, where signals are then relayed to the muscles of our hands and the other muscles in our body. And when the device is turned on, it generates this magnetic field illustrated here that passes through the skull into the cortex and is able to stimulate electrical activity in the neurons underneath. And that can generate action potentials in these motor neurons that travel down the axon to the muscles and then cause contraction of the muscles. And so it's this induced movement of the body through stimulating neurons in the brain. One limitation of this technique is that it's generally more useful for studying the function of areas of our brain that are close to the surface of our scalp. different areas of the cortex, and particularly at the top or the back of our head. It's not so useful usually for studying structures located deeper in our brain. Another limitation is that generally it's not targeting the function of any one particular neuron. And so we saw that with this, when we have electrodes that are inserted into the brain, we can place those electrodes in contact with one specific neuron and manipulate the activity of that neuron in order to learn about what its function is within a particular circuit, for example. Whereas with TMS, because we're only applying the stimulation from the surface of the head, we end up stimulating sort of a whole chunk of the brain that's located near where the device is placed. So it's giving us this less precise control over the activity of neurons. Another related technique is called transcranial direct current stimulation, and this is very simple. It's where electrodes are placed to the surface of a person's head and electrical current is passed between them. So again, this is a non-invasive technique. So it's not, the electrodes are not being inserted into the brain; they're just placed on the skin, and it's seemingly safe under the right conditions. And to give you an example of how it might be used to study cognition, so one recent study illustrated here used this technique to apply electrical stimulation to around the kind of prefrontal area of the brain at the front here, so illustrated in red here, and then applied this stimulation while people were training how to do this difficult working memory task. And they found that people who were practicing this working memory task while receiving this stimulation ended up learning more and getting better at it compared to people who just practice the task but without the stimulation. And so, again, a feature of this technique is that it's not targeting any one particular neuron. It's really targeting kind of a diffuse area of the brain, and that limits the kind of research questions that we can address using this technique. Okay, so the key points here are that changes in brain function, whether that's due to damage to the brain or sort of temporary stimulation of the brain, are able to provide insight to the brain mechanisms that underlie cognition. And these brain stimulation techniques vary. Sometimes they're targeting individual neurons, but that comes at the cost of having to kind of open up the skull to insert an electrode. And sometimes these techniques are just causing sort of more diffuse manipulation of brain activity. Okay, so final section before we have a break, we can now talk about different methods for recording the activity of the brain while a person is performing some kind of task. The first method we've come across before in this course. So in lecture 2 on the action potential, I began the lecture by talking about this study that inserted an electrode into the brain of cats, recorded, and that electrode would come in contact with one specific neuron at a time in the cat's brain. So this was in the occipital cortex at the back of the head. And then they showed the cats different images on screen, and in order to sort of test what kind of images would produce activity in this neuron. And what this data on the right is illustrating is that for this one particular neuron that they're recording from, when the cat is looking at a vertical line, that neuron is producing lots of action potentials. When it's looking at a horizontal line, that neuron isn't responding. And so this technique is providing really specific information about the activity of one particular neuron. So the data that's recorded would be the voltage of the cell over time as that changes over the course of milliseconds, and that allows us to visualize the action potentials that are happening in the neuron that we're recording from. In humans, again, this technique has been used at times, and we'll see an example later in the course, but generally it's used in animal research instead. But we can record the electrical activity of the brain in humans using less invasive methods as well, and one is called EEG, or electroencephalography. This involves placing flat electrodes on the surface of a person's head and recording electrical activity just from the surface of the head. And it turns out that all the kind of action potentials and electrical activity of the neurons in our brain together is sufficient to cause electrical activity that can actually be measured from the surface of our body. And so this is what EEG is doing. And so the signal that's recorded might look something like this, where in the y-axis is the voltage that's been recorded, fluctuations up and down in voltage. In the x-axis is time. And this technique has good temporal resolution, so similar to single cell recording. So it's able to measure changes in voltage from millisecond to millisecond and describe the time course of how electrical activity is happening in the brain in detail. But it also has relatively poor spatial resolution. So to illustrate what I mean by that, in the case of single cell recording, the electrode is coming in contact with one single cell, and the data that we're recording are the action potentials of that particular cell. In the case of EEG, because the electrode is just placed on the surface of the head, it means that the signals that it is picking up, the electrical activity, won't relate to any one particular neuron in the brain, but rather will reflect the joint activity of maybe thousands or millions of different neurons. Okay, so if we zoom in to the part of the brain just below one particular electrode, we'll find a chunk of cortex, and in that cortex there will be maybe millions of different neurons, and the signal that is propagating through and being detected by the electrode will in some way reflect the kind of the summed activity of those neurons. So it's not recording individual action potentials, it's just recording this sort of net electrical activity of a whole group of neurons. Despite that, it's still useful for different types of research questions, and so it's good for tracking broad changes in activity that happen in our brain. So we'll see in a later lecture that when we fall asleep, there are these kind of wide, sort of large-scale changes in the activity of our brain, and even throughout the course of a night, like when we're dreaming and things like that, there are these sort of broad changes happening in our brain activity, and these can be identified quite well in this kind of signal that's been recorded by EEG. Another example is epileptic seizures. So when a person has a seizure, there tends to be this kind of chaotic activity of neurons across the brain that sort of spreads from one area of the brain across to neurons located elsewhere. And that activity shows up very kind of vividly in this EEG signal. If we want to identify where activity in the brain is happening a bit more precisely, we generally need to use different techniques compared to EEG. And one technique is called positron emission tomography, or PET. And so PET involves, first of all, injecting a person with glucose that has been modified to contain radioactive atoms that emit gamma rays. And so glucose is the main, or one of the main energy sources in our body that our body extracts from our food. It's also referred to as blood sugar, and so it travels around our blood and supplies the different organs of our body with energy. And importantly, when a part of our body is more active, it consumes more energy, and that occurs in the brain as well. So when a part of our brain is active, there will be more blood flow and more supply of glucose to that and other resources to that area of the brain. And so what that means is that once somebody's been injected with this radioactive glucose and they're performing some sort of cognitive task that's engaging a particular part of their brain, there will tend to be greater uptake of that radioactive glucose in that more active part of their brain. And then the PET machine is able to detect the radiation that's being emitted by the radioactive glucose and infer where in the person's body or where in their brain the glucose is being more concentrated. So some examples are shown here, where somebody who is like watching a silent movie has this, the PET machine is detecting a greater concentration of this glucose in the back of their head at the occipital cortex, so supplying the neurons there with more energy because they're becoming more active as a person watches a movie. When a person is listening to somebody speaking, there tends to be more activity on the sides of the brain in an area called the temporal cortex. And when somebody is thinking, like they're giving a difficult maths problem to solve or something like that, there tends to be this greater activity at the front of their brain. Okay, and so the idea here is that we're visualizing brain activity not by recording the activity of neurons directly, but rather tracking this sort of indirect signal, which is the supply of glucose to different parts of the brain. A limitation of PET is that it involves exposing people to a dose of radiation, and it involves these specialized chemicals. A technique that's become more widely used in research nowadays is functional magnetic resonance imaging, so fMRI. And so earlier we talked about MRI being used to generate anatomical images of the brain. And in that context, it's referred to as structural MRI. But an MRI machine can also be used in a different way to measure changes happening in activity of the brain over time. And in that context, it's referred to as functional MRI. fMRI involves measuring something called the BOLD signal, so the blood oxygenation level dependent signal. And it effectively relates to, again, to the supply of blood to different parts of our brain, which is being used as a proxy of which areas of our brain are becoming more active. And so it's based off the principle that when neurons in a part of our brain are becoming more active, producing more action potentials, the blood supply tends to increase to that area of the brain over time in order to supply those neurons with the resources that they need. So including glucose, but also oxygen. And so the technique, you don't really need to know the physics of it, again, but to give you a sort of a general picture of how it's working, it depends upon this protein hemoglobin, which is found in our blood and has this particular role in carrying oxygen around our body. So it carries oxygen from our lungs through our bloodstream to the other organs of our body, including the brain. And hemoglobin reacts differently to the magnetic field that's generated by the MRI machine when it has an oxygen molecule attached to it, compared to when it's given up that oxygen molecule to a neuron or something like that. And because of that, the machine is able to measure a signal that relates to the level of oxygenated hemoglobin in different parts of our brain, and use that as an index of where the blood and resources are being sent in our brain. And ultimately, this leads to a picture of brain activity that has relatively good spatial resolution, so it can track kind of where those changes in blood flow or oxygenation are happening in the brain within a few millimeters or so. But the temporal resolution is not as good as if we were measuring the activity of neurons directly, if we were measuring the actual action potentials or electrical activity of neurons. And so what I mean by that is that we can create pictures of which parts of the brain are receiving more blood flow and which we infer from that are becoming more active, but we can't measure like the number of action potentials that a neuron is producing, for example, precisely. Okay, so there are actually thousands of studies now that have used fMRI to try to understand the role of different brain regions in cognition. Just to give you an example of one study, so you have a sense of what the data looks like. Here they were, again, looking at this kind of emotion recognition task that I described before, but now they were testing healthy controls and measuring the bold signal using fMRI, the flow of blood in their brain effectively, while they were completing this task. And in particular, they were comparing the bold signal between trials where people were looking at images like this and judging the emotional state of the person versus trials where they were looking at the exact same image but judging the age of the person. And then they compared activity on those two trials in order to try to isolate brain regions that are specifically involved not just in looking at an image of a face, but in making these judgments about a person's emotional state. And what they found is illustrated here, these different areas of the brain that are becoming more active in this task. Okay, so this is just a table from the textbook in case it's helpful when you come to revising the content of the lecture, comparing the different techniques. The key learning points here are that there are these neurophysiological methods that study the function of the nervous system. These, again, can be targeted towards the function of individual cells or can be targeted more towards understanding how activity is happening across the brain at a kind of whole brain level. Some techniques measure the electrical activity of neurons directly, recording action potentials, for example, while others are measuring these metabolic changes like the supply of glucose, the supply of oxygen to different parts of the brain as a kind of indirect measure of neuron activity. And as a consequence, some of these techniques have better temporal resolution, they're better at describing how the brain is changing its activity over time, whereas others have better spatial resolution, they're better at describing where in the brain changes are happening. Okay, so are there any questions at this point? Okay, so before we have a break, we can do just a couple of poll everywhere questions. And so the first question is to imagine that a research team is interested in mapping the network of brain areas that are involved when people solve maths problems. They want to visualize which areas of the brain are active as people do this, as they do maths in their head. Which method would be most suitable for this study? EEG, fMRI or PET, microelectrode recording, so single cell recording, or electron microscopy. Okay, so so most of the class is saying B, which I agree with. So these techniques, fMRI and PET, are good at specifying sort of where in the brain which areas are becoming active, and so this will be useful for mapping out a kind of network of areas that are becoming active when we're doing some kind of cognitive task. EEG is not quite as good at defining where in the brain activity is happening. Single cell recording in a sense has very good spatial resolution that we can, if we've inserted an electrode like 1 centimeter into the occipital cortex, and we know exactly where the signal is coming from. But it's generally only recording the activity of one cell at a time, or if you have multiple electrodes, still a sort of small sample of cells. And so it's not going to be very good for kind of looking at the brain as a whole and mapping out a network of areas at once that are active. And then electron microscopy is this technique that's used primarily for producing anatomical images of cells. Okay. So next question. So a neurobiologist wants to understand the precise role of a single neuron in a rat's motor cortex as it's moving. They plan to stimulate activity in this neuron and see what effect it has on the rat's behavior. Which method would be most suitable? Transcranial magnetic stimulation, transcranial direct current stimulation, electrical stimulation using an electrode that's been implanted into the rat's brain, or pharmacological stimulation, like feeding the rat a stimulant like caffeine or amphetamines or something that upregulates or stimulates brain activity. Okay, so again, almost all of you put C, which I agree with. So an electrode that's inserted into the brain is able to modulate activity of a single neuron at a time in order to understand the role of that neuron in a particular function. These A and B are describing these non-invasive stimulation techniques that apply either magnetic or electrical stimulation from outside of the head, and as a result they tend to be modulating the activity of larger chunks of the brain, and so not very good at understanding the role of any kind of one particular neuron. And then this is sort of analogous to some extent with pharmacological studies, where we might know that a particular drug can influence neurons, like inhibit neurons that are releasing GABA in our brain, for example, or something like that. But if we take that drug and it's in our bloodstream, it will be delivered all over our brain. And so again, it's not really helping us understand the role of any one specific neuron. Okay, so let's have a 5-minute break and come back a bit before 10 past. Thanks. Okay, so let's get started again. So in these last sort of 20 minutes or so, I'm going to try to give you an overall picture of the anatomy of the nervous system. And the idea here is to make you familiar with the kind of structures that exist in our nervous system and the kind of terminology that's used to describe the brain, so that later on in the course, when we talk about a particular brain area, like the amygdala being involved in emotion or something like that, you have a bit more context of what that area is in the wider picture of the brain. To start with, we can divide the nervous system in general into the central nervous system, which is made up of our brain and our spinal cord, and the peripheral nervous system, which includes motor nerves, which are used by our brain effectively to control the muscles of our body, sensory nerves, which are carrying signals from the body to the brain, and then the autonomic nervous system, which is shown in red here, which refers to nerves that are extending out from our spinal cord and our brain to control the different organs of our body. A spinal cord is this sort of column of neural tissue that's in our back, protected by the vertebrae of the spine, and it acts as a kind of relay station, I guess, between the body and the brain. We're looking at a cross-section here of just like one particular part of the spinal cord, and you can see that there are nerves that are extending out of the spinal cord, carrying signals between the central nervous system and the peripheral nervous system. And then there are also signals being conveyed up and down the spinal cord as well. If we look at a cross-section of the spinal cord, perhaps the most obvious feature is this sort of darker region of tissue on the inside and the lighter region surrounding it. That darker region is called grey matter, and it refers to tissue of the nervous system that's made up primarily of cell bodies of neurons. And then that lighter tissue is called white matter, and it's made up primarily of the axons of those neurons. A key anatomical structure in our peripheral nervous system are the nerves, and a nerve is effectively a bundle of axons, and so it's where the axons of a bunch of different neurons are forming together into a pathway, if you like, that travels from our spinal cord, for example, out to some other part of our body. Some of these nerves also have a structure called ganglia, and these are these little kind of lumps that you see in the nerve here. They're most commonly found in sensory nerves, and they are formed by clusters of cell bodies of the neurons that are making up that nerve. The autonomic nervous system refers to these nerves that are extending out to interact with different organs of our body, like our heart, our lungs, our stomach, and so on. It's often separated into two halves, two competing halves. One is the sympathetic nervous system, which has a role primarily in preparing our body for fight or flight. So when we need to act urgently, we need some of our organs to be most active. We need to upregulate our breathing, our heart rate, and so on. Whereas the function of other organs, like our stomach, might be less crucial to our survival, and so we downregulate the activity of the stomach. The parasympathetic nervous system has the opposite role of preparing our body for resting and digesting. So quieting down the activity of our heart, for example, and upregulating the activity of our stomach. So the key points here are that the nervous system can be divided into the central and peripheral nervous systems, where the latter includes this control of autonomic functions of our body, so the vital functions of our body. And then our nervous system, the peripheral nervous system, has these particular anatomical structures. It has nerves and it has ganglia, and these are formed by the axons and the cell bodies of neurons. Before we talk about the brain, it's useful to understand different types of views that we can have onto the brain. And so generally we're going to be looking at the brain using particular cross-sectional images that expose the different structures found inside the brain. And these cross-sectional images are generally taken along three main anatomical planes, which are referred to as horizontal, sagittal, and coronal. And so a horizontal cross-section of the brain is where we've cut the brain like this, and we're looking kind of down on the brain or from underneath. A sagittal cross-section of the brain is where we've cut the brain sort of around the midline, along that axis, and then we're looking at it from the left or the right. So this is the front of the brain here and the back over here. And then a coronal section of the brain is where we've cut it like this, and then we're looking at it from the front or from the back. And that allows us to see the left and the right hemisphere of the brain. There are different terms used to describe regions of the brain based on where they're located relative to each other. A very simple distinction is between left and right. So our brain is made up of two hemispheres, and we refer to that as the left and the right hemisphere of the brain. Another distinction is between dorsal and ventral, where dorsal refers to the top of our brain, ventral refers to the bottom of our brain. So I always remember this by thinking of the dorsal fin of a shark. So if you're swimming in the ocean and you see a fin coming through the water towards you, then that's the dorsal fin of the shark. It's on the back of its body. Another distinction is between anterior and posterior, which refers to the front of the brain, where our face is, so that's anterior. And posterior refers to the back of our brain, the back of our head. And then finally, we have lateral and medial, where medial refers to the center of our brain along the horizontal axis, and lateral refers to the sort of left or right sides of our brain, the more peripheral sides of our brain. So to put that into practice, here we're looking at the left hemisphere of a person's brain, and highlighted in red is a region called the prefrontal cortex. And so we refer to this as being the left prefrontal cortex. If we then look at the medial surface of the left hemisphere, so the right hemisphere is missing and we're looking onto the brain from the right, then we can refer to that as being the medial prefrontal cortex here. And then we can split that into a dorsomedial prefrontal cortex, the part that's located closer to the top of the head, and a ventromedial prefrontal cortex located closer to the bottom of the head. And so that's the medial prefrontal cortex. Similarly, if we focus on the other side of the brain here, that would be the lateral prefrontal cortex. And again, we can define a dorsolateral prefrontal cortex and a dorsoventral prefrontal cortex. Sorry, dorsolateral and ventrolateral prefrontal cortex. Okay, so the key points here being that there are these anatomical planes that we often use to provide a perspective onto the brain, and in particular there are these sagittal, coronal and horizontal views of the brain that we will generally be looking at. And then there are these anatomical terms of location that indicate the relative position of different brain regions, and in particular the main terms are dorsal, ventral, anterior, posterior, medial and lateral. When we look at a cross-section of the brain, one of the most sort of obvious anatomical features, again, is this distinction between darker and lighter areas of brain tissue. And just like in the spinal cord, we can refer to this as white matter and grey matter, where the white matter is made up primarily of the axons of neurons, whereas the grey matter is where the cell bodies are found in greater density. Some of the grey matter we can define as being nuclei. And so if you look at the brain here, you can see that there's grey matter around the edge of the cortex, but then there's also these lumps of grey matter deeper in the brain, and these are the nuclei. They're clusters of cell bodies of neurons in the central nervous system. So these are analogous to the ganglia that are found in our peripheral nervous system. And these nuclei can often be linked to different functions and given different names. And so as an example, this lump of cell bodies that you see here, this grey matter, is called the caudate nucleus. Last week we talked about the neurotransmitter serotonin and how it's produced in these neurons that are found deep in the brain in these structures called the raphe nuclei. And so these raphe nuclei, again, are just lumps of cell bodies of neurons, and those neurons will be sending their axons out through the white matter of the brain to release serotonin across different parts of the brain. And at the beginning of the lecture, we talked briefly about the subthalamic nucleus, the STN, which again is a lump of grey matter deep in our brain that has been linked to the voluntary control of movement. And so you don't need to remember the names of these particular nuclei. I'm just trying to illustrate the concept of this particular anatomical feature of the brain that we find, and how these nuclei can often be linked to different functions. The white matter of the brain is also organised. It's organised into different tracts. And so a tract is a bundle or a pathway formed by axons in the central nervous system. So this is analogous to the nerves of our peripheral nervous system, but a sort of pathway of axons in the brain is referred to as a tract instead. And so that technique, MRI tractography, that was visualising the axon pathways in the brain is visualising these tracts of white matter. Some of these tracts have names as well. So, for example, there's one called the corpus callosum, which is labelled here, which is really distinctive because it's connecting between the left and right hemispheres of our brain. So the key points here are that grey matter is where the cell bodies of neurons are mainly located, while the white matter of the brain is formed mainly by the axons of those neurons. And we have these anatomical structures, the nuclei and the tracts, which are analogous to the ganglia and the nerves that are found in the peripheral nervous system. Okay, so a key part of the brain that we'll talk about a lot during this course is the cerebral cortex, because it's linked to many kind of important aspects of cognition. It's the largest part of the brain by volume in humans and other mammals. So when we're looking at a view of the brain from above here, everything that we're seeing is the cortex. If we look at a cross-section of the cortex, what we find is that the very top layer of the cortex is this thin sheet of grey matter with all this white matter underneath. And so you can think of the cortex as being like a big sheet, a big thin sheet of grey matter that's then being kind of scrunched together in our head, forming all these sort of wrinkles and folds in its surface. Okay, so the idea here is that in that grey matter, there are cell bodies of neurons, and many of those neurons are projecting their axons out through the white matter in order to communicate with other neurons in the brain. The cortex has these wrinkles and folds, and their proper name are gyri and sulci. Okay, so a gyrus is a peak or bump in the cortical surface, and these are separated by the sulci, which are these depressions or grooves in the cortical surface, these wrinkles in the surface. Sometimes we can also define fissures, which are like a long and deep sulcus, and so the most obvious of those is something called the longitudinal fissure, which runs down the top of our head and separates the left and the right hemisphere. And when you look at a picture of the brain like this, at first glance it probably looks pretty random and messy, all these wrinkles, but it turns out that this folding of the cortex is actually consistent enough across people that we can label particular gyri and particular sulci that will be found in pretty much everyone. Okay, so all of us will have a central sulcus, a groove that's sort of running down our cortex on each side of the hemisphere here, and then on either side of that central sulcus there will be a gyrus that we can refer to as the precentral gyrus and the postcentral gyrus. The cortex is divided into four main areas that we can refer to as lobes. So the frontal, parietal, occipital, and temporal lobes. And those are found in both hemispheres. The occipital lobe we've touched on already and seen that it's critical for our vision. So in the occipital lobe, there are neurons that are receiving signals from our eyes, processing those signals to help produce what we see. The temporal lobe is found on either side of our head. It also has a role in vision. So, for example, we'll see in a later lecture that there are neurons here that are receiving signals from the occipital lobe and involved in processing more complex visual information, like recognizing a particular person's face and things like that. The temporal lobe also has a role in hearing. It's also receiving signals from our ears. The parietal lobe also has a role in vision, so quite a large part of our brain is just dedicated to vision. The parietal lobe also has other functions, so for example, perception of our body. When we feel something touching our skin, signals are being sent from our skin up to the top of our head, the parietal lobe. And then the frontal lobe at the front of our head tends to be linked more to kind of higher order cognitive functions, like our ability to plan, to predict the future, to control the movements of our body, and aspects of memory and emotion as well. So overall, the cortex is involved in perception, cognition, and action. The very front of our cortex is referred to as the prefrontal cortex. And many of the anatomical features that we've talked about are conserved across different mammals. So all these different mammals will have an occipital cortex, a temporal cortex, and so on, a frontal lobe. But one feature that many people think distinguishes humans from other mammalian species is this expansion of the prefrontal cortex that occupies a larger kind of chunk of our brain. And the prefrontal cortex has been linked to these executive functions of the brain: decision-making, working memory, and the kind of intentional control of our behavior and our attention and our emotions and so on. Okay, so the key learning points here are that the cerebral cortex is this folded sheet of grey matter with white matter underneath. It can be divided into four main areas, the four lobes, and it's involved in sensory processing, control of movement, and different cognitive functions. One broad division of the brain is between the cortex, which is taking up much of the volume of our brain, and everything else. So the structures that are making up our brain that are found sort of deeper and located underneath the cortex. And we refer to those as being the subcortical structures of the brain. So that includes the cerebellum and these other structures highlighted in yellow here. To point a couple of these out, the cerebellum is this dense brain area located at the back of our head underneath the occipital cortex. Traditionally, it's been linked to the control of movement based off the observation that people who have suffered damage to this area often lose coordination. They can become clumsy and find it difficult to control the coordinated movements of their body. More recently, there's been more and more research linking the cerebellum to aspects of cognition as well, like learning, for example. But it's still a relatively kind of murky area of the brain where we're still figuring out what it does and why it has so many neurons as well. The brain stem is this deepest structure of our brain that connects between the spinal cord and the rest of the brain. It's made up of these two structures called the pons and the medulla, illustrated here. And you can think of the brainstem as being like the autopilot of the body. It has this key role in regulating the vital functions of our body via the autonomic nervous system. And so that includes the control of our breathing, our heart rates, our sleep, and so on. And so these are things happening in our body that most of the time we're not paying any sort of conscious attention to, and we sort of often have limited conscious control over them, but they're still being regulated by our brain through the brain stem. The thalamus is found above the brain stem, up here, And we'll see later in the course that this acts as a kind of relay station between our senses and the cortex. And so the thalamus is receiving signals from our eyes, from our ears and other senses, and then it's relaying those signals to different parts of the cortex. And pictured at the bottom here are these different tracts of white matter that are connecting the thalamus to different parts of our cortex. And then a couple of other subcortical structures that we'll talk about later in the course are the hippocampus and the amygdala, which are these structures that are located deep in the temporal lobe in each hemisphere. And we'll see that that the hippocampus is linked to memory, particularly the formation of episodic memories. And the amygdala has been linked to control of our emotional responses, like fear, for example. And then one final structure to mention before I let you go are the ventricles. So when we look at a post-mortem brain like this, something that stands out are these big holes in the brain, and those are actually meant to be there. So we all have these holes in our brain, but when we're alive, they're filled with this fluid, which is illustrated in blue on the left here. And that fluid is called cerebrospinal fluid. It's found in these ventricles within our brain, these kind of reservoirs of fluid within our brain. It's also found around the outer surface of the brain and also around our spinal cord. And it plays a role in cushioning the brain and supporting the weight of the brain, and also seems to have a role in helping to kind of wash away waste products from the brain that are created by neurons, and perhaps also supply nutrients to neurons as well. Okay, so the key points here are that there are these subcortical structures of the brain, and some that you should be aware of are the brainstem that has this role in coordinating the vital functions of our body, the cerebellum that's been linked to movement as well as other functions, the thalamus that acts as a sensory relay point, and these structures within the temporal lobe involved in cognition, like the amygdala and the hippocampus. Okay, so any questions? Okay, so of course you can come up and ask me questions now if you prefer, and there's just a few more tables at the end here from the textbook to help with your revision. The tutorial running in week 5 and 6 will be based off anatomy and help you kind of consolidate some of what we've covered here in a bit more of a hands-on way. And next week's lecture will talk about vision in the brain. So thanks, I'll see you then.